and career transition awards, as well as seed grants to support this very important work. So please join me in welcoming Dr. Patel.
Hi, everyone. My name is Kunal Patel. I'm one of the neurosurgeons here at UCLA, and one of the scientists. My lab is just across at Gonda. Today, what I'm going to hopefully show you is how to marry my clinical passion with my scientific passion, and how one helps the other, and then the other helps the former. We're going to talk a little about two things, actually. I just added a second thing because I'm a new member to BRI. I've been a faculty here for 2 years. I'm looking for more collaboration. I'm looking to just show the community what the types of problems that I'm working on as a clinician and as a scientist. And I'd love to get feedback from you guys. Please, I'm gonna have my email at the end. Like, this is more so you guys get a bird's eye view of what we're working on in the neurosurgery department, and see if it, like we were talking about before, sparks any interest. There are two problems that we're going to talk about today. The first is brain cancer, and the second is a condition called hydrocephalus, where you can't drain the fluid, the cerebrospinal fluid that you make in your brain, out of your head. We're going to start with glioblastoma. Glioblastoma we're not doing a good job still. The national progression-free survival is very poor. The overall survival remains very poor. Here at UCLA, or at academic centers, there have been studies saying, okay, with our clinical trials, with specialty surgery, with specialty radiation, specialty chemotherapy, good quality care, we still only get to about 20 months. And, even starting here, and I do most of the glioblastomas here at UCLA, we are struggling to get even beyond that. Okay? The FDA-approved targeted therapy, right? That's the whole idea behind doing science on glioblastoma, has led to very few FDA-approved medications and with poor survival benefit. So what is the main problem? From a surgeon's perspective, this is what I think the main problem is, that it is an infiltrating tumor. This cancer is your brain. So these are some pictures of my patients. This is a type of tumor called a meningioma. This comes from the covering of the brain. Everyone here can see the normal brain, right? Beautiful, and then the tumor, really ugly. This is a lung cancer metastasis, right? So, cancer from all over the body can spread into the brain. Again, you can see this looks like the lung. This is the brain, right? And this is me. I can find the barrier between the two and say, okay, I'm going to remove this. This is brain cancer. It is the brain. You can't see where it starts and where it ends, okay? And guess what? I can't see it either, all right? And so it's not just you. How I've dedicated my career, or what I would like to do in my career, is systematically target infiltrating tumor. And we do that in 3 ways, or I'm thinking about doing that in 3 ways. 1, being able to see it. 2nd, being able to understand it, and then using that information to really develop novel treatment strategies. So these are 5 patients that I did this last year. These are big tumors here, 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 and here. I resected them, and then the radiologist says, you resected them, and like, great, good job. But what I've noticed is on standard imaging, which is right here, it looks like you removed it all. See, the white is gone. But on different types of imaging, you can see that there's abnormalities at the depth of the resection at the bottom. I can't tell the difference between the normal brain and this abnormality. And that abnormality always predicts where the tumor comes back. And so what I worry about is that that is actually tumor that I'm leaving behind. It's very hard to see in surgery, and it's hard to see on standard MRI. This is what we call non-enhancing tumor, versus all this stuff, which all of us can see, is enhancing tumor. This is the infiltrating tumor. This is the tumor that's inside the patient's normal brain that's functioning well. And so what I worked on when I was in training is with Dr. Ben Ellingson and Jingwen Yao, who are both faculty here in the Department of Radiology, is a pH-based method for identifying tumor. We were originally doing this in traumatic brain injury, and then what I noticed was it looked like what I thought was left over after surgery. So there's the main red here, and then around it is kind of the infiltrating tumor. So now we have a pipeline where we can see the contrast enhancing tumor, which is this, and then using this PHMRI, look at what might be the infiltrating tumor. And in fact, this technique does, with good sensitivity and specificity, identify infiltrating tumor. We can look at the tumor on H&E after I remove them in surgery, and you can see solid amounts of tumor, infiltrating tumor, going 50% tumor, 50% brain, and then 0% tumor, all brain, like at the margins. And then most importantly, if I'm able to resect the non-enhancing tumor to the point where the deep biopsy is negative. Okay, it looks just like normal brain. I'm not getting recurrence. Okay, and so I've only done this in like three to five patients or something like that. It's only been a couple of years, but like I said, these people were getting progression-free survival in 4 to 6 months, and now we're talking, at least these three people, it's been 27 months. And so it's a dumb idea, but it's actually, which is just remove the whole tumor. Okay. But what I think we need to do this is basically adjuvant, or really go to the lab, whether it's the computer lab or I'm going to talk to you in the basic science lab to try to figure out how to actually do this. And so we have a trial that's almost open, and it's a phase 1 using these pH-related techniques or MRIs to resect the tumor. These three patients, or the patients I showed up here, are just the perfect candidates for these resections. They're in the areas of the brain where you're not really getting any functional deficits. Many patients don't qualify for that. So how do you actually target this tumor in cases where you can't actually remove all of the tumor? Because it's going to cause the patient to be paralyzed or blind or not able to speak, or which is called aphasia. So what we do now is, with Dr. Kornblum, you all know, is do these biopsies for all of our patients where we take the contrast enhancing or solid part of the tumor and the non-enhancing part of the tumor. And we sequence it, we culture it. We published this paper together when I was training in Dr. Kornblum's lab, where we took infiltrating tumor and started characterizing this using single-cell, single-nucleus sequencing. More recently, what I've been trying to do is understand how functionally different these invading tumor cells are. And so I grow them in gliomospheres, and then with Dr. Aparna Baduri, grow them in her organoid, a cortical organoid model. And are noticing there are differences in growth rates, differences in how they attach, and this may be a functional difference in how these cells are working versus what I can remove in surgery, right? I can remove the solid part of the tumor in surgery. I can't remove the infiltrating part. That's what's left, and that's what needs to be treated medically. In addition, our standard chemotherapy, which is this temozolomide, this is the standard of care in the United States, the blue, works not even that well in the solid part of the tumor, but what I'm noticing is in these patient samples really don't work in the infiltrating tumor. The top is about 48 hours, the bottom is 2 weeks. And so really it takes long-term chemotherapy to actually get effect. The thought here is that perhaps they're slowly cycling cells in the infiltrating regions. And this is something that we're looking into with Dr. Kornblum. And so the last thing is how to actually treat infiltrating tumor. One of the things that is a major problem in our field is the blood-brain barrier. In samples from my patients, I looked at blood-brain barrier proteins, and in infiltrating regions, you can see the astrocyte foot processes around the blood vessels really well, versus in the solid tumor, it's all broken down. And that makes sense to me, at least as a clinician, because the contrast that we inject into the patients leaks into the contrast enhancing region, which doesn't have a blood-brain barrier, but does not leak into the non-enhancing tumor because it has a blood-brain barrier. But for treatment purposes, that makes it really difficult to get drugs to the infiltrating region of tumor, which again, as a surgeon, is the problem. And so what we're working on is this is what a resection cavity looks like. So this is like 3 months after surgery. This is what it looks like. So there's a big hole here. It's connected to the ventricle, and there's a lot of unique pharmacodynamics that occur here, right? If you can't get drug into the infiltrating tumor through the blood, one of the thoughts is to try to get it directly into the resection cavity. And so one of the early or preliminary stuff we're doing is looking at pharmacodynamics of chemotherapy agents in a resection cavity, because the pressure in this cavity can vary patient to patient. How much fluid is flowing through the cavity into the ventricles can vary patient to patient. And so this is one of the things that we're working on as well. I want to pause there and just give you a brief overview of a second leg or part of our research program, which has to do with these resection cavities and what's called the ventricular system. So again, I do a lot of these GBM resections. After the resection, there's a resection cavity. It's connected to this black structure here. This is the lateral ventricle on an MRI scan. And what I notice is and classically the teaching is if you enter this, you have a risk of hydrocephalus. Hydrocephalus is an inability to absorb spinal fluid. We make about 300 cc's a day, which is like a amount of like a soda can, and it needs to be absorbed. When it doesn't get absorbed, then it builds up in the head, and it can be very dangerous to patients. So one of the things that I do to fix this problem is when I'm in there, this is a surgical view where we are actually using these yellow coagulating, they're called bipolar coagulation, to coagulate the choroid plexus without hitting any of these arteries here that would cause the patient a stroke. And that actually has shown to do a couple things. One, we did MRIs prior. The red here is a segmentation of the choroid plexus. And then after the cauterization, showing that the amount that I can take about, I can decrease the choroid plexus volume by about 50% using that cauterization technique. And then we have dropped our hydrocephalus rates in patients where we enter the lateral ventricle from about 25%, which is the standard in the historical standard, to like 5%. And so I think that this choroid plexus cauterization helps from a clinical perspective. And so this was a purely clinical project. What we did was we said, well, if we're cauterizing it, can we actually remove it? And so what I started to do after IRB approval is I cauterize this, but I just cauterize the stem of the choroid plexus, and then I cut it. And so now we can actually sample human choroid plexus for the first time. Human choroid plexus cannot be sampled traditionally because this is in the middle of the brain. And so it really can be only done, like we're really taking advantage of this opportunity to look at this organ, which we really don't know a lot about. We know classically it's involved in cerebrospinal fluid production, but all the studies are in animals or post-mortem because you can see how complicated and how uncommon it would be to be able to get choroid plexus. So I've been doing this for the last couple of years, and we have no safety issues with this. And we are able to get basically in all samples good quality human choroid plexus. So afterwards, I cut out the cauterized portion on the back table because it's not going to be good tissue and good specimen. And then we started by just looking at H&E. These are H&Es of the choroid plexus with the intact anatomy. And then we sequenced it. So we sequenced now 10 human choroid plexus samples and from people of different ages. And so we used a cutoff of greater than 60 or less than 60. I don't do pediatric patients, so I don't have that data, although I have a partner who we're hoping to also get from like large tumor resections in children down the line. But what we noticed is there are changes to the choroid plexus over time. And specifically, when you look at the epithelial cells, this is the inner lining, this is the cell type that is supposed to be producing the spinal fluid. They cluster together very well by age. And over time, all of the transporters known to produce cerebrospinal fluid are decreased as you age. To try to test this, so in addition to sequencing, we tried to grow the choroid plexus cells as like a human model and see if they can actually produce spinal fluid. And so we use this two-chambered model where we put the choroid plexus epithelial cells, which are the cells that are supposed to be producing this fluid, from the patients on this in this model. And then we put fluid and fluorescent dextran in both of the compartments. We're right now benchmarking this model, to be honest, making sure that there's actually like a electrical resistance between them, like that it actually produces a barrier, fluidic barrier. But what we've noticed is, yes, well, at least not in all samples, but in our best kind of culture model, we can have it prevent any electrical movement of fluids. And what we notice is using the fluorescent dextran, that the epithelial cells do produce a small amount of fluid. We need to characterize this fluid, but my thought and hope one day is that this is actually a model of producing cerebrospinal fluid. There are a couple receptors here that have been studied in like the 90s in animals and in monkeys, but really hard to translate to humans because you don't really have a human model. And so we looked at all of those previously studied transporters, and it really came down to sodium gradient. And it's similar to if anyone did physiology with the kidney, except for the reverse. And so all of the same transporters that you see in the kidney that reabsorb sodium and then reabsorb water, so then, to produce urine, it's the same, or it's a similar type of mechanism where you're first pumping sodium across the gradient and then water is falling through aquaporins. We knock down one of those receptors, and we can see in our model that that the rate of fluid production decreases. And so this is one of the things that I think we're going to be working on over the next few years, benchmarking this model, understanding human choroid plexus, both from a bioinformatics standpoint and a functional standpoint. And then overall, what my lab is trying to do, as I'm about 50% clinical, 50% research, is using the problems that I see in the clinic. And for me, specifically, it's brain cancer and this condition called hydrocephalus, and using what I have access to, which is human tissue, to then use in the lab and guide how I do surgery. Like now I do these choroid plexus cauterizations, and how I hopefully one day will treat patients. I'm definitely in need of collaboration, ideas. And so if there's anything that you guys have sparked your interest in this talk, please reach out to me. I think it would be really fun to do. The choroid plexus is really a black box, and now that we can open this, I mean, the amount of stuff we can learn from this is huge, because it's not just spinal fluid. It's neurodegeneration, it's Alzheimer's. Like there's so many different combinations. So if there's anything that's interesting and you want to look into the choroid plexus with me, I'd love to work with you. Thank you, guys.
Thanks. So first time I've shown these results outside the lab. I kind of threw them together in the last like 3 days since just kind of heard about this talk last week. So they're a little sloppy, but hopefully you guys will get something out of it. So as Lindsay helpfully said, I'm currently affiliated with Freed Lab in what we call the Cognitive Neurophysiology Lab at UCLA, and I previously worked at University of Pennsylvania, and this is actually a collaboration between these two labs. Here's Freed Lab here. There's a few of these people in this room, so that help record data from patients and help set up tasks and things like that, work with surgeries, and Dr. Freed there is in the middle, who's quite well known for research in this field. Particularly human intracranial single unit recordings. That's Mike Kahana, my previous postdoctoral advisor, who actually designed the task, that's the sequential task I'm going to be talking to you about today, along with Chris McDonald of TimeCell fame when he was at UPenn. And Daniel Schoenhout is a was a PhD student with Mike that developed the initial version of what we'll call the goldmine task. And I'll be mentioning him quite a few times since he's kind of the inspiration for a lot of these initial studies. Daniel was actually a UCLA undergraduate. That's his picture in the Daily Bruin way back then. So what is neuronal replay? There's a lot different talk from the last one, but I'll try to take you all through it. And so it's this pretty classic finding at this point that was discovered probably a few decades ago in rodents, where if a rodent runs through a linear array, so you'll get place fields. So everyone kind of knows what place fields are at this point. There are cells that fire at specific places. There's been a Nobel Prize won for them at this point. And you can see this is 13 different neurons, and they'll tile the kind of running of the rodent. So you'll get these neurons, 13 neurons firing in this sequence. And what's fascinating is that after the rodent stops running and then is usually, eating behavior, or sorry, eating or doing some sort of like quiet behavior, you'll see these right after the run, you'll see these reverse replays. These are zoomed in here. So you can see this is over the course of maybe 50 milliseconds, these neurons will recapitulate their firing in the same sequence. In this case, in the reverse order. They also do this, and this paper, this is from a paper that's been cited 2,000 times. This is like a very famous finding at this point. And we also see forward replay. The forward replay actually tends to occur, either when the rodents are about to do a behavior. Here you can see this forward replay was shown to occur before the rodent ended up running down the track. And again, you see this sequential firing of neurons in a row. This was initially discovered in sleep. It's more recently been shown to occur during awake behavior. And so more in like quiescent states when the rodent's being kind of quiet. And again, this is like a very popular field. I mean, there's thousands of citations on these review articles. So why is replay important? So there's been a couple very recent reviews about awake replay in particular, and off the clock but on the job. And so the new theories on kind of what awake replay is doing is, instead of online decision making in rodents, the idea that this is helping with the decision that's going to happen right now, it seems that awake replay performs an offline fictive learning for later goal-oriented behavior. So the idea being that when the replay helps to recapitulate these experiences and helps you learn, so then sometime in the future, whether it be in another minute or in another couple hours, you're going to have something, you're going to have learned something, and you have practiced this thing so that you can use it for future behavior. And even more kind of esoteric is replaying computational computation from several people at DeepMind and UCL, is that replay has implications for AI systems, which lack the human ability to radically generalize past experience to solve new problems. So the idea is that this thing I just described, being able to rehearse things to learn and then solve problems, is something that AI can't do right now. And so it's thought that replay might be how we help to solve this. So have we seen it in humans? I've talked to you, it's very well cited in rodents, as indicated by the citations. And first I'll talk about a few methods just before I get there, because in humans we really just have intracranial methods. So intracranial methods look a lot like this at UCLA. This is at Cedars-Sinai in early Rudishauser's lab with a patient that obviously was kind enough to give image rights. And I don't really know what they're doing with their fingers here, but so we don't do that. But we put a laptop in front of people, then we have them run tasks. In this case, the tasks I'm going to show you today are virtual reality tasks. They're really just navigating on a laptop and using a mouse to kind of navigate in this environment on a laptop. We use intracranial recordings. So Dr. Fried places electrodes for clinical purposes only, but these patients selflessly agree to put in research electrodes that transverse the hollow lumen of the research electrode. So this is all a research electrode. And then these microwires will come out the middle into these medial structures. So we can actually record some number of single units in humans. Dr. Fried really helped popularize this technique. The electrodes are called Benke-Fried electrodes. And now back to, have we seen it in humans? So Karim Zaghloul was a neurosurgeon at NIH. He was able to do some recordings in anterior temporal lobe. This is more of a outside structure or superficial structure. And he found these sequences, just like we just saw in rodents. We see these sequences of neurons firing in a row. They happen during encoding. They see these sweeps that happen right after the person first sees a pair of words. And when people retrieve the words, if you give Jeep, they have to then say crow. These events, these sequences really start to ramp up right before they say the thing. So there is some indication in humans that we see these. So why are they able to get these beautiful recordings? Because this is tissue that was going to be resected. Karim was able to put some Utah rays, jam them onto this tissue, I think in the OR, do some recordings with the patient, and then proceed with the resection. So these are superficial structures. This is not the classic hippocampal replay we think of, because that replay is thought of as being very memory-related medial tumoral things and associated with hippocampal ripples. This is maybe something different, but it's very cool that the brain seems to have these conserved mechanisms that seem to be occurring here. In particular, that what this replay seems to be doing in a subsequent Nature paper he published was that you can decode categories with this. So if you have a sequence that looks like this for birds, like a crow, it'll show up for other birds. And if you then try to reorder the cells for automobiles, they won't look the same. But there will be separate sequences that do automobiles, and they'll all look the same for automobiles. So it's very semantically related. So it's cool that these exist, but I think this is really something different. So this is one thing that's been done in humans. Really, the only other paper is from Fried Lab last year, again, a Nature paper. But really, this wasn't the main point of the paper, but there's some very cool findings here, and this is really inspiration for what I'm going to show you today, is that, so I'll take the time to go through the task a little bit, is people see sequences of images. They just have to do a simple mirror task to say, is this the image or a mirrored version of the image? A very implicit, very boring, just kind of a rote behavior task. But there's an underlying structure to the presentations where the red image will always be followed by the green image, the blue, or this image here. So they have to follow the edges of this graph structure. So for a long period in the task, when the patients are learning the task, blue will never be followed by green. Patients don't actually explicitly notice this, but they pick it up over time, and they learn. And what's really interesting is that Pavel, the later author of this study, found that if you actually look during the rest periods, because you take some rest to do this task, it's very laborious. In these rest periods, the patients would kind of have a minute to rest and they'd start the task again. As they kind of learn this implicit structure, if you start looking at little short windows, he used 30 milliseconds in this case, in these rest periods, you would start to see the neurons replaying the order that you actually see from the task. So green, red, blue would show up more often than green, blue, red, indicating that there's some kind of sequential replay going on. And these are hippocampal and entorhinal, in this case, neurons. And they do seem to be firing in sequences, and they seem to be happening in these interstitial periods when a person's kind of in the crux of learning the implicit structure of this task. So getting at some of the stuff we talked about in these reviews earlier, I'd say for the first time in humans, was in this paper. I'm hoping I'll convince you that I've seen something quite like this, and in a pretty exciting way, because I have some very nice control conditions I'm able to use that Pavel did not. So Goldmine is this task I mentioned that Daniel Schoenhout published in 2023. This was a task really designed to look for time cells. And in this task, so it's going to be really important to kind of remember what these task periods are. So I'll try to really be explicit about them, and I'll try to reuse kind of the figures throughout so you can kind of be reminded. But people navigate in this virtual maze. This is what the view looks like when they are actually doing the task. They start in this base, and then they are told to go find 4 pieces of gold that are kind of scattered throughout the maze. They do this during the gold search period, which comes after an initial delay period where they just kind of stand in the initial home arena. There's another delay period where they stand in the home arena. They're told to kind of visualize the path they're about to take to go, and then afterwards they have to go retrieve the gold they found. Then the gold's hidden, but they go up and they press the space bar. And if it's there, then it'll show up and they'll get a score. So the idea of this task is you really want to separate time cells from place cells. So so this is what a place cell looks like in a human. This is, the maze. This is an overlay. You can see the cell during both the searching period, also we call it the encoding period. You see the cell fires down here, and the digging period fires kind of in the same area. So this place cell kind of resolutely encodes some sort of place in this, just like it does we see place cells in rodents. He was also very interested in time cells, really designed the task for this. So what you see is the time cell is something that just kind of fires at the same time every time across trials. There's 36 trials of this task, and you can see it resolutely fires here, pretty much at time 0 of the task right at the beginning, but only at the search period, not in the dig period. So unlike the place cell, it doesn't really seem to map the behavior to both. This was actually a major finding in this paper, that actually these place cells were relatively invariant. They would fire the same in these 2 navigation periods of the task. The time cells would remap. They would largely fire specifically only in their periods. This will become relevant later because I'm going to show you some kind of neuronal replay related to these specific cells. So that's Goldmine. Now the task I'm really going to focus on today is GemMine. Goldmine ends up becoming control because Goldmine doesn't necessarily have any sequential behavior. But GemMine was this task designed by Kahana Lab. I wasn't involved in the design process, but I've been quite involved in the analysis process, where in the middle of the task, between the searching period and the delay period, and now we've changed it to gems. So now it's just not just gold. It was always gold before. Now here's a sequence of 8, we're going to put 4 gems in the maze on each trial. Could be out of these 8 gems, it switches for every trial. And then the participants are asked in the middle of this trial, place on a timeline reflecting the 30 seconds of the gem search period. We want you to place where you found each of the gems when you found them. So a very sequential order task, a little bit like that Simon thing I was showing earlier, if anyone remembers that. And they, of course, also have to place the correct gem identity, and they're picking it from trial to trial. It's not an easy task. I've done it. It's hard. There's a lot of interference trial to trial, so it is very challenging, which is good because then people get better and better at it, and I'll show you some behavior in just a second. But there's really only 2 differences in the task. One is we put a timeline period in the middle, right before the second delay, where they have to do this kind of task here for points. And the second is we use gems instead of gold. Those are the only 2 differences. Everything else is basically the same. So here's some behavior. So focusing on this digging period where they have to go find the gems. Again, the gem's identity is hidden, but they have to kind of go back to the same spatial location, press dig, and they're shown this icon here if they got it correct. They are penalized if they get it wrong, so they aren't just running around dig, 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 dig. And so this is number of correct digs per trial. The maximum they could possibly get is 4. And in general, yeah, participants get better over time. There are some participants that don't really get the task very well. They only maybe get 1 on average, even in the 3rd tercile behavior. So there's always 36 trials and dividing into terciles. I'll say for the neural analysis I'm about to do, cell pairs is kind of the currency I'm going to go through. And out of 400 cell pairs, only 2 of them came from these 3 participants. It tends to happen that way where the participant is not doing the task very well, then neurons won't necessarily be very active. So it does tend to happen that way anyway. But yeah, for the most part, pretty much all of the data I'm going to show you is coming from participants that are improving at the task and are pretty good at it. What about the timeline period? So this period here, we can also assess how well they did at this part of the task. And indeed, they improve during the task. And yeah, they do pretty well. They get something like, they begin at 60%, they get up to 70% correct over the course of the trial. Correct being that they put it in the correct order and on the timeline. This is the average error. So remember, they're supposed to put this is supposed to reflect 30 seconds on this bar. So we can actually measure the error in seconds of how close they got to where they actually first viewed the gem. It actually does better than where they actually picked it up. So when they first view it, you can see, yeah, they generally get maybe a little bit better, but yeah, they're somewhere within like 5 seconds. So in general, participants doing well the task. Okay, so a quick little interstitial part about neurons. So remember, the gold mine task, they have to find gold. They're told, if you have 3 more gold to find. Here's the recordings we have from humans. This is kind of like what we can get from 10 participants: 85 hippocampal neurons, 100 entorhinal neurons, 92 amygdala, et cetera. In GemMine, we ended up getting exactly the same number of hippocampal neurons across 12 participants. Just for completely random reasons, we didn't get many entorhinal neurons or many amygdala neurons, or as many prefrontal neurons. It just kind of happens where sometimes you just don't get participants with the right electrodes. They're totally chosen for clinical reasons, the placements. And also maybe just, I think Fried was souring on the task a little bit because no one was really looking at it. And I think once you see some of these results, and maybe we'll watch this on YouTube later, he's going to be pretty excited, I think, about a lot of the results here. And I've already kind of showed him some of these. And so, yeah, hopefully we'll get these tasks done in the early stage of the patient stay, because the neurons tend to fade over the course of the patient stay. So what's interesting is if you just take these neurons and do the simplest analysis, one of the simplest analysis you can think of, which is just take the 2 delay periods, the rest periods, and then take the 2 navigation periods, and just do a t-test between them. So average across trials, do we see neurons are selective to delay or selective to navigation? In the goldmine task, the regional task that didn't have the timeline period, yeah, there's a fair number of delay and navigation selective neurons. In the GemMine task, where you added this temporal component, where they still have to go and they still have to retrieve things in space, but now there's kind of the task is a bit different, where they sort of are told to remember sequential things. Suddenly, the navigation neurons in hippocampus largely disappear, at least selective ones. The delayed neurons are very active. So even this little manipulation, just gems plus timeline, it really seems to change the behavior a ton in this task. I think goldmine is really a navigationally driven task, and I think this task people are really thinking about sequences a lot more. And as for my title, you'll see that that's kind of meaningful. So I'm going to go over methods real quick. These methods are pretty important. So I'll point out that we can't look at forward and reverse replay with the number of neurons we have of this order. I would need 13 neurons in a given participant. You probably need 100 neurons in a given participant. I'll point out, though, the first couple of papers that showed this kind of sleep replay really just looked at pairwise neurons, just like I'm going to show you today. These papers, have been cited thousands of times, are seminal at this point. It really wasn't until 2006 that David Foster was able to get enough neurons. And they could get 126 neurons in the hippocampus of one rat. In our patients, we can get on the average of something like 8 neurons per participant. So this is why I'm limited to just kind of showing you some pairwise replay results. But fortunately, I do think it's very convincing because of the fact that we have a couple tasks we can compare. A quick PSA about methods. If you want to look at pairwise replay between 2 neurons, you have to normalize their firing rates. If you just take 2 neurons and these are simulated neurons, I'm just doing some simulations. If you take a 5 hertz neuron, a 7 hertz neuron, you say, okay, well, how often does the 7 hertz neuron fire within 50 milliseconds in the short window we're expecting replay within the 5 hertz neuron? Well, of course, there's more spikes in the second one, so it's gonna look like there's forward replay. It's fake. So you need to correct for this. Same thing if you have a 7 hertz neuron, a 5 hertz neuron, and you look and say, how often does the 7 hertz fire and then the 5 hertz? Well, it's going to look like reverse replay because it's always going to look like the first neuron's going to have more spikes than the second neuron. So this is kind of the nitty-gritty method part, but basically I need to subsample spikes. So anytime I have 2 neurons with different firing rates, I subsample spikes from the one with the higher firing rate to match them, to make sure I am not seeing fake replay. So obviously with simulations, you can correct for this. Another key part is some neurons are more bursty than others. So here's 2 simulated 7 hertz neurons. You can see the second one looks a lot different. Neurons fire bursty a lot of the time. So to correct for this, what I'm going to do is for every pair of neurons, and every time I subsample, I then run a permutation test where I circularly shift the neurons. So I take one of the neurons and I shift it in time. So now it's kind of a fake time neuron that's not locked to anything. And then I run the my whole pairwise replay analysis and say, do I see neurons that fire within 50 milliseconds? And basically gives me just a basal rate of what do we expect by chance to happen between these 2 neurons, just based upon their firing characteristics between every cell pair. So what I'm going to show you on the y-axis and all these plots coming up is what I call baseline subtracted replay. I'm going to subtract out the basal level of what these neurons should expect to replay by chance. More explicitly, for each cell pair, the proportion of spikes of one neuron occurring within 50 milliseconds of the other above chance levels. And finally, I'm just going to tell you that I'm going to look at forward and reverse replay, just like they do in rodents. It's a little weird to be like pairwise forward reverse replay, but the best I can do is really I can say, well, if I have 2 cells that fire, and one of them fires during this the search period, this encoding period, if one of them fires at time 0, the next one fires at time 3 seconds, well, a forward replay event we would expect would be 0 to 3. 0 to 3 would be forward, and if the neuron fired, the 3-second neuron fired, and then the 0-second neuron fired, that would be more like a reverse replay. So I'm basically just taking the search period and I'm just averaging across the period and saying, when does this neuron peak its peak in firing during the initial exploration, when they're first kind of learning the stimuli. And some results do come out from doing this, and I'll show you them in a bit. All right, so take a breath. We went through the boring methods just to try to convince you that I'm thought about this quite a bit, and it's really important. I've only been doing this for about 3 months now, and first month was just kind of figuring out how to correct for methods. Man, I didn't get a single laugh on the Full House thing or the Saved by the Bell thing. So on the results, a lot of the plots you're going to see right now are going to look like this, so I'll explain this in detail. I have 412 cell pairs in hippocampus across my participants. Obviously, I have to look at cell pairs within the participant, for it to be meaningful. And I'm going to be looking at within 50 milliseconds, do we see cells firing one after the other in these very short little windows. Every dot here is a cell pair. Every period in the task, this is every period of the GemMind task I'm showing you here in order to try to keep everything aligned. Every period here has 412 dots because, I can just look at the same cell pair in every single period and compare the periods. And again, everything on the y-axis you're going to see is baseline subtracted replay. So how much replay do we see above chance? And what's fascinating is right off the bat, just taking every hippocampal neuron I have across all my participants that have at least, one cell pair. And right away, you start to see these levels rising during these delay periods, just like you would expect in a rodent, and also during the timeline period. So during the period when people are not navigating, but they are placing these objects on the timeline. And then in the encoding and the retrieval period, these 2 navigationally based periods, you just don't see as much replay. I'll point out that the black dots here so each pair here is the forward and then the reverse. So every swarm here this is the forward replay, this is the reverse replay. You can see a little hint of increase of reverse. I'm going to be able to pull that out by using some cell types in just a second. But every single plot you'll see it's forward then reverse for each of the 5 periods. And as you can see, I kind of use this interchangeably. Encoding is the search period when they're first looking for the gems. Retrieval is when they're then trying to find the gems afterward. And I'm going to compare all this now to Goldmine. So I can do all the same analysis in Goldmine, all the same thing I just told you about. But again, Goldmine doesn't have this kind of sequential component to the behavior. Or at least it doesn't have to. People could use sequences. They could try to do it in the same order. But as you can see, when you take all the hippocampal cell pairs in Goldmine, 602 cell pairs across participants, you see very little evidence of the same replay you're seeing during these delay periods. There is no timeline period in Goldmine, so I can't compare that. But it's just kind of almost at zero. You see very little evidence of this. So these are the first 2 points I'm going to make right off the bat. So first, human replay is stronger in periods without navigation. So just like in rodents, we're kind of recapitulating this effect, even when they're just using, doing navigation on a laptop. And I think what's really interesting is it's in periods of little perceptual versus internal conflict. So even in the timeline period, this is something we don't really have in rodents, where they can just sit there and place things on a timeline. We also see replay during that period. I don't think something like that has quite been seen before. This kind of awake replay during an actual behavior, as opposed to just quiescently sitting and planning planning your next move or thinking about your last move. And then replay occurs when sequences matter. So I'll keep showing more evidence of this, but it really seems like in the task that people were required to do sequences, this is what really brought out the replay, at least in humans. I don't know if anyone remembers. This was a game where you had to kind of memorize sequences and things like that. And I remember it being quite hard. Okay, so now I'm going to run through a lot of these slides really quickly and show you things. First thing, we don't get a lot of contralateral recordings, but in rodents, they show that these sequences will cross ipsilateral to contralateral. You'll get neurons on both sides. I don't see that in hippocampus with these participants. So again, this is the same plots you just saw on the top. Let's keep showing these same ones for comparison. The contralateral side, I just don't have many participants where we had contralateral recordings in hippocampus. So I'm just going to keep showing ipsilateral for the rest of the talk. What about if we take those delay selective neurons? So early on, I showed you this pretty staggering difference where it seems like in GemMind, these delay selective neurons are really important as opposed to the navigation selective neurons. So if I take my cell pairs only by delay selective, it cuts my cell pairs to about a quarter of the cell pairs. Now you really start to pull out some differences. So when you take these delay selective neurons, these are really the ones that show this sequential, this pairwise replay during these periods. And again, encoding, retrieval, you can see the distribution is just totally different. It is particularly driven by one participant who actually did the task twice, but there are some other participants that showed some data. We do hope to collect more data on this task. So if you do the same manipulation with Goldmine, take only the delay selective neurons, you start to pull out a little bit of replay. So it's not like it's totally absent in this task compared to GemMind, but it's not nearly as strong. And here you can see the participants are a bit more balanced in terms of number of neurons recorded. But still, in that encoding retrieval period, you see nothing in Goldmine. If you take the remaining neurons, it's pretty flat across the board. So it's really these delay selective neurons. The neurons that fire the most during delay, well, they're firing the most because they're the ones that seem to be active and kind of doing whatever this processing is that happens during these delay periods, both in rodents and apparently in humans. And also in Pavel's paper as well in humans. So this is kind of the third big point. I'll get back to that in just a second because I'm going to show you some it's going to be part of the third point, is the fact that what if we look at some other regions? So I've been focusing on hippocampus because this is where replay is classically seen in rodents. It's been shown in entorhinal cortex. We see sequential replay in entorhinal cortex. I don't really have enough participants in GemMind to see that, unfortunately. I don't really see much in the little bit of recordings I had, just 16 cell pairs though. In Goldmine, we happen to have more. You see maybe a hint of it, a bit more in delay 1 and delay 2 than coding retrieval, but it's maybe about the same as hippocampus and Goldmine, but again, Goldmine doesn't have the sequential task, so we really just need more participants, and we'll certainly try to get them next time we have some entorhinal cortex coverage. Amygdala, here I did have a decent amount of cell pairs, and again, you just don't really see that many neurons. I'm selecting for only the delay selective ones, so I'm kind of whittling down my population, but not as much evidence of this happening in amygdala and prefrontal cortex. Again, I don't have a lot of participants. There has been some kind of sequential replay shown in prefrontal cortex. I'd really have to have more participants to tell. So right now, I'd say still, I'm not seeing anything at the level of this kind of 5 to 7%. So what this kind of intuitively means is that above baseline levels, I'm seeing for every spike in the first neuron, 5 to 7% of the time above levels, I'm seeing a spike in the second neuron. So it's a pretty impressive result to get these kind of numbers up in this range. And we're not seeing this in the other regions. So point 3, replays seems specific to hippocampus and occurs in a delay selective group of neurons. Okay, so now I can get into cell types a little bit. So I'm going to show you similar plots, but now, just like in the goldmine task, a big part of Daniel's task was to find time cells, find place cells. We can do that in GemMine. Again, we find place cells, 12% cells in hippocampus show up, or actually these are all cells, but we see them a lot in hippocampus, show up as place cells. And then really the time cells and the time phase cells, remember I told you earlier, they're specific. They fire specifically to encoding or specifically to retrieval. So that's what time phase means, especially in the interaction. So yeah, we find a decent amount of time and place cells. And I can basically replicate that figure from Daniel's task. So he showed, this invariance in the place cells that doesn't exist in the time cells where between the search and the dig period. I see a similar thing. Place cells are pretty invariant, maybe not quite as much in GemMine, but again, this was just about significant. So I see cells that act a lot like they do in goldmine, where they act like time cells, they act like place cells, just like we see in rodents. And now what I can do is I can compare and I can say, okay, well, time cells, I expect them, if they're going to be sequentially firing, I expect them to fire in order. So if you see a cell that's the model's time 0 and time 3, we expect it to fire 0, 3, 0, 3, 0, 3. If it's a forward replay or 3, 0, 3, 0, 3, 0, it's a reverse replay. Place cells, it's a little harder to interpret, but you can basically just say from the beginning of the maze, that's going to be what a lot of the place cells are for initially, to the outside of the maze. So you expect the order to recapitulate encoding. So we expect the place cell, if it's forward replay, to fire here, and then towards the outside. So I'm just basically defining how I'm defining my forward versus reverse replay. And the reason for that is because, well, time cells, it's not the most amazing effect I've ever seen. There's a bit of a, you see an enrichment in the population and kind of the timeline and delay periods. But what's pretty interesting is that when you took at the place cells in GemMine in particular, you start to see this enhancement in reverse replay. So even though it's like I'm kind of using these methods that are a little, I can't do as well as the rodents can with the methods and really kind of pin down with all the neurons, even with this simple assumption that I just assume that the place cells in the beginning are going to fire, should fire before the ones near the outskirts of the maze. Well, what happens is in rodents is at the end of a run, you see a lot of reverse replay. That really seems to be what I'm seeing in participants as well, that in these delay periods in particular, in the timeline period, after a run, you kind of see these reverse replays, almost like, but specifically for the place cells. Okay, so just a few more slides. You might wonder, okay, well, you've shown pairwise replay. It's not the best evidence of showing these beautiful sequences they can see in rodents, but can you at least do something like triplets? Can I see 1, 2, 3 in a row, 3 neurons firing in the same sequence? That gets into a bit more of the sequential thing. And it's really only a couple of participants I can show this in that had enough neurons to be able to show sufficient triplets, but you do see evidence of this where I can see the proportions are going to be lower because this is conditional on the fact that if there's a spike in the first neuron, do I see them in the same order for then 2, 3, 2, 3. So 1, 2, 3, 1, 2, 3, 1, 2, 3. So you're always taking the first neuron as kind of like what defines the proportion. So what proportion of spikes lead to a sequence? So the proportion of spikes of any given neuron that lead to a sequence is something on the order of 3% or something like that. But it is stronger in delays in that timeline period again than in encoding retrieval periods. Goldmine, I just don't see anything. So really when you conditionalize on triplets without the sequences in the task, they really just disappear. So I'd say this is, in a sense, so this is replicating kind of the first and second points I made earlier, but I just want to stop for a second and just say this is pretty cool because in rodents, I can't think of anything like this. Rodents, they just see replay. Rodents, the simplest thing they do is they run on a linear track and you see replay that occurs. I don't think a null result like this has been shown where you show in one task versus another task that there's a lot of replay in this one task and there isn't replay in the other task. So this is something very human in nature that it seems like these mechanisms are not necessarily always going like they are in rodents, at least as far as we know in rodents. Okay, so last little bit is, do we see replay over trials? You might be wondering, as people get better at the task, like I showed you earlier, both they dig better and they find more things, and they also do the timeline period better. In Hippocampus, we see the replay grow across terciles. So as participants get better at the task, it seems like we see, particularly in these delay periods, we see more and more replay. So this is starting to get this idea that when they are learning, the replay is helping them learn. I'll show more evidence of that in a second. Goldmine, you just don't really see the same thing. Again, Goldmine, it just doesn't seem like this sequential learning. I don't remember why I did this slide. Oh, this was the time cells and place cells. Sorry, my thing's blocking it a little bit on the side there. But interestingly, if you do look only at the time cells, you do see this evidence again. Sorry, I switched the periods here. I didn't really mean to. I didn't have time to remake the slide. The delay in the timeline periods, again, you still see with these time cells, this kind of incremental during learning. Weirdly, in the place cells, those ones that just seem to recapitulate past experiences in rodents, they don't show this increase with learning, almost indicating a separation here between what time cells and place cells are doing. The place cells just recapitulate this past thing. They don't necessarily involved in learning. The time cells do seem to be involved in some way, or at least they show this sequential increase in replay with the learning. So point 4, replay improves throughout the session as participants improve at the task. So it's pretty cool. There's not a lot of evidence in rodents of this. There's maybe some that it's kind of like you kind of impart rodent behavior onto the rodent. Very last thing, you might be wondering, okay, well, if this replay thing is really happening, then you should really be able to say, if replay is happening during this delay period, and that's going to help influence behavior, like we hypothesized in those beginning review articles, then we should see the replay in particular on a given trial inform the behavior on that same trial, but later on, when they're actually doing the behavior. So here, I'm going to look at this delay period here, and then we'll say, okay, do they dig more trials when you see more replay in the trial before? So simple model, delay to replay, correlating with number of correct digs. Here's a plot to help explain it. So if the person's about to get 4 right, how much replay do you see? And I put it in that bin. If the person's about to get 0 right, well, you put the amount of replay on the y-axis here in this bin, and you can see a direct correspondence. If you put it in the XFX model, it's very significant. It does seem that on a trial-to-trial basis, the more replay you see, the better they're going to do with behavior on that trial. Very similar idea. You can just do the timeline period only, this mystical period where it's not really a rest period. They're actually doing behavior. If you correlate the amount of replay in the timeline period versus how often they place the things correct in the timeline, again, a direct correspondence between, if they're going to get 4 right in the timeline, you're going to see a lot more behavior. Again, very significant. As a control, I could look at the encoding period and say, okay, well, does encoding replay, where we didn't really see a lot of it, but what if I take it and see what I did see? Does it predict either of these behaviors? And it does not. You see it's not significant for either of them. And that's it. So increased awake neural replay on given trial improves future behavior. So this is something that's, has been hypothesized in a lot of these review articles. They look at some rodent behaviors and say, I think we're seeing this, but it's kind of hard to say. I think this might be some of the first time we're able to show this because we're able to do it with, human behavior. I tried typing that last sentence into ChatGPT, and it did a pretty good job of what I wanted, because I kind of said a futuristic, world, but then it just drew a bunch of tadpoles in the person's brain, so it still needs some work. So I'll just point out again that quote I said from earlier: instead of online decision making in rodents, because again, all their studies have been done in rodents at this point, awake replay performs offline fictive learning for later goal-oriented behavior. I think I'm seeing something of that sort, where during these delay periods, when people are learning and figuring out how to do things, it leads to the subsequent behavior doing better. Future directions in 5 seconds. I have several papers on hippocampal ripples. So there's a big kind of white whale, or I don't know, black, or whatever the expression is for what I haven't done, blind spot. I need to do that. You can see in all these things I haven't shown before. These sequences that happen in 50 milliseconds. The reason I use 50 milliseconds is about as long as ripples get in humans. I will look at that soon. And then because all these events in rodents are couched in these hippocampal ripples. And like I said, we need more data. We really need to record some interrhinal and prefrontal, and probably another really good hippocampal patient like that one we have to really pin this down. And that's it. Thank you to a lot of people. And Itzhak Fried for, I run his kind of the day-to-day operations in his lab, and things are going well. When there's funding, we'll see how things go in the future. But and also a lot of big thank you to our selfless participants that do this to no help of their own, and just kind of do this because they want to contribute to society. So it's very heartwarming. Thank you.